Okay, AP Bio, this is part 3 for chapter 37 here. And after this, we've only got one more chapter, maybe a few little things to end with, but very tiny things if we even do that. So you would have seen this image up on Jupiter by now and taken this simple little quiz. And what I want to do now that we've talked about an action potential is move on to putting a bunch of action potentials together to make a nervous impulse or a nervous system or nervous tissue. And one of the things that we need to deal with is what happens in between the impulses, and that is the refractory period. It is a tiny bit of a time where the sodium gates are closed completely and cannot be reopened, but it is a limit. It's a limit to reaction, it's a limit to stimulus in the senses, it's a limit to memory, it is a limit and upper cap on our nervous system. And so what happens is that when the sodium gates close and they won't reopen, that causes the next gates to open in one direction, one way, and that will move the impulse in a certain way but not allow it to come back. If the impulse could come back, then our nervous system would never be able to communicate with anything else inside the body. And so this is not a limitation or a weakness, it's built in so that we can move impulses down a neuron, down the axon, and basically along a series of neurons all the way to the brain and back. So the action potentials, when they shut down for one, when they do the undershoot, the sodium gates open up for the next part of the on the membrane for the next part of the axon, and the sodium gates open, and then when they go through their process, the 1, 2, 3, 4, 5, and then those close, and the next ones open, and on we go, generating an action potential down a nerve cell. You could imagine what it would be like if it came back or went in two different directions at the same time. That would be a state of confusion in your body and in your brain, and you could never conduct a signal long distance. And so the conduction of action potentials starts in the axon hillock, right at the base of the nerve cell. It is an electrical current generated by the movement of ions in and out of the membrane. But everything hinges on those sodium channels being inactivated, closed for just 1 to 2 milliseconds. And that means that the impulse, the wave, the synaptic wave that you saw in that video moves from the nerve body, the axon hillock, all the way down to the synaptic cleft. And that is how you have impulses moving all which ways in your body. You just have to aim the neuron in the right direction because the impulses don't travel backwards. Now, the speed of an action potential of which this happens is based upon, has a direct relationship with the diameter of a neuron or a nerve cell. Invertebrates have the ones that are fast, like the squid that you dissected, they have really large neurons. You can see them with the naked eye because they're very fast and very active, but they have to have large neurons to pull that off, much like your book says, like a garden hose. The larger the hose, the more water you can pump through it. But vertebrates have really tiny nerve cells, and that's because they insulate them basically with fat. It's a myelin sheath. Certain glial cells produce lipids around the neurons that don't conduct electricity, and they only have the gates, the ion gates, at the junctions in between. And so that nerve impulse will skip or move or jump very quickly down the neurons. So we are high tech. We have tiny little wires that are insulated. The impulse moves even faster than it does in invertebrates, all because we insulate them. Now, on a personal note, my grandfather died of a type of cancer. He was advanced in age, but still, he died of a type of cancer that ate away the myelin sheath around his neurons. And basically what happened when the neurons came in contact with each other in his brain, it would cause seizures because that impulse would jump every which way. It wouldn't flow down the neuron in the direction that it was supposed to. It's pretty rare, but it did happen in my own family. And so that's the importance of having insulated neurons. It's a really advanced structure only found in vertebrates. By now, you should have watched the video of how synapses work with I think a question or two at the end. That is the whole puzzle put together, from the chemical structure to the ion flow to the individual nerve cell to stringing them together. And if you're able to put these pieces together of how neurons work at the ionic level, at the cellular level, and then when you string them together, you are on your way to studying what a nervous system is. So neurons communicate with each other. Yes, they have electrical impulses where a current flows, but they communicate with each other chemically. And so we have a neurochemical system, to be truth be told, and we are coming full circle to that cone snail. And so the chemical issue happens at the synapse. There's a gap. None of your neurons are actually touching each other. And so at the postsynaptic junction, at the synapse, at the cleft, there's a cell, another neuron waiting for it, and you don't have electricity that shoots across the gap. You have chemicals that shoot across the gap. They will then trigger reactions in a new neuron, which will send an electrical impulse down its body. But you guys really need to stop and think about this, what it means for illicit drugs, prescription drugs, alcohol, and all kinds of things that are put out there, from OTC stuff to just stuff in our culture, is that you have a neurochemical system. The cone snail knows this, and so do other people out there, whether they be pharmaceutical companies or doctors or, anyway, bad people, I guess. And they know that these chemicals will interfere with the nervous system and can cause all sorts of side reactions. And so, anyway, a lot of these normal neurotransmitters, like acetylcholine, they are repackaged, reused, recycled. Some of them are removed through hydrolysis. They're just broken down, washed out with water, basically. But most of it is reused and recycled because it's really expensive to make these things. And so, anyway, make sure you know. Really green and blue, repackaged, recaptured is the primary issue. Removed is usually pretty rare unless it's an intense situation like an adrenaline rush or whatnot. So every neuron has connections with, could be thousands of other neurons depending on where it's located and what it's used for. But these neurons all have different effects, and so you're going to learn about excitatory and inhibitory neurons. And so they will all send just an electrical impulse, but they don't all mean stimulate. Some of them mean calm down or relaxed. And so the cell body of one postsynaptic neuron may receive many inputs from hundreds or thousands of other neurons with their terminals touching it. And so a single neuron is not enough to really excite a large stimulus on another neuron. You have to have hundreds of neurons fire at the same time for your legs to coordinate, for your brain to think, for your heart, to pump at a regular rhythm. And so we have different types of ways of decoding what these neurons mean. So if you get two excitatory neurons coming one after another, that's called a summation. And that will just mean that the stimulus is pretty strong, but that's about it. It has to do with frequency. You can have a temporal where one jumps on top of another right after it, and then that goes ahead and fires off an action potential. You can have them come in at the same time, or you can have an excitatory, then an inhibitory, then an excitatory, and they kind of cancel each other out. That's called a spatial summation as well. So if you look at this graphic, you'll be able to see a number of ways that we can start decoding a message. All of this is done through the neurotransmitter acetylcholine. It's the primary one that's used in both invertebrates and vertebrates. We make it, we manufacture it inside ourselves. And all of our receptors in our neurons are built to receive acetylcholine. So sometimes, not always, but sometimes you can have states of depression, chemical depression at least, because you don't make enough neurotransmitters. And if that's the case and it's that easy, then it can be chemically corrected. It's not always that way, but sometimes it's that way. Now, I think this is fascinating. Acetylcholine is used for muscle stimulation as well as memory formation and learning. In other words, students, moving, memorizing, and learning all go together. I have no idea why you sit in a desk all day and try to learn really hard things, but I wish you guys could work out in the mornings beforehand, or stand up or walk around while you learned, but that's not our culture. Anyway, you can have a number of things interfere with these. Botulism, as you can see there, it comes from a gas that bacteria make, and people will inject this into their facial muscles to relax the muscles, to quote unquote get rid of wrinkles. But basically, there are far more neurotransmitters than that. And depending on what you do with them, you can induce all sorts of reactions into people. So in your book, they have this graphic where they're showing you the chemical structure of some of the major ones that you should know at the AP level. I'll talk about them bit by bit, but if you're savvy in chemistry or looking to take AP chemistry or college chemistry or something like that, then you're going to need to know and be able to recognize these ring structures and chain structures for these biogenic, these neurotransmitters like the biogenic amines, amino acids, and gases. So if you've ever gone to the dentist and they put nitrous oxide on your face, laughing gas, to relax you, and of course you've heard of terrible things with carbon monoxide leaks in homes or in the garages or whatnot. These are not stored in your body. They're incredibly dangerous because they're basically inhibitors for the peripheral nervous system. But they are produced in your body. And if you want to know why they're produced, the reason they're produced is basically because life is painful. And they're used to relax or block pain in the body. So your body will naturally produce these and then it'll get rid of it. After that, you're going to read about some biogenic amines. They're related at the core structure of the molecular structure to the amino acids. They're not amino acids, but they do have that component, that functional group. And so epinephrine is a pain reliever, pain reducer, and then you have dopamine and serotonin. Those are pleasure chemicals. We'll talk about them more in the brain function in the next chapter. But your body releases those to suppress pain, to reduce stimulation, and because life is painful sometimes at a biologic level. So, but they're also used for learning, which we'll talk about in the next chapter. So the one that a lot of athletes are very familiar with are endorphins. They mimic morphine. You might have heard of the runner's high or been super relaxed or feeling good after a workout. These are endorphins. They hide pain as well and discomfort. They don't last very long in our body. The problem with the opiate crisis in our country is that it mimics these endorphins in a synthesized level, and they are highly addictive because it works so well. So at the end of your chapter, they have this little graphic of these different types of toxicities or concentrations of synthetic versus natural pain relievers, basically. And you can start to learn why you have to have higher concentrations of the synthetic ones, just because they're not keyed to the body. So that is it, gang, for chapter 37. I am going to have you turn in a scientific skills exercise for 37 and 38 at the same time later. So if you've listened to this lecture, done all your reading, and uploaded your notes for chapter 37, that's it. Talk to y'all later.